Cancer is a disease that begins in the cells of the body. Normally, human cells grow and divide to give rise to new cells as the body needs them. When cells grow old or become damaged, they die, and new cells take their place. When cancer develops, however, this orderly process becomes disrupted. Old and damaged cells survive when they should die, and new cells are formed when they are not needed. These cells can grow and divide uncontrollably and give rise to tumors. Medulloblastoma, a type of cancer originating in part of the brain called the cerebellum, is the most prevalent type of brain cancer in children. This cancer accounts for 18% of brain tumor formation, leaving children with a rate of survival as low as 30%. Growth and division of cells is coordinated by precise molecular machinery known as the cell cycle. The cell cycle is tightly regulated by checkpoints that serve to ensure proper conditions are met before the cell replicates its DNA and divides. These checkpoints are controlled by cyclin-dependent kinases, CDKs, which are proteins that bind to their cyclin partners. If growth conditions are unfavorable, the cells will arrest at the checkpoint. If conditions are posing no harm to the cell, CDK will bind cyclin, allowing for progression through the cell cycle. In my lab, we study a cell cycle regulatory protein called SPEEDY, also known as SPY1. SPY1 binds certain CDKs and is able to override select cell cycle checkpoints, thereby providing an avenue for cells to overproliferate and become tumorigenic. Interestingly, SPEEDY is implicated in the maintenance of the expansive and therapy-resistant stem-like populations of brain tumor-initiating cells in the most aggressive brain tumor known, glioblastoma multiforme. Therefore, it is my hypothesis that SPY1 drives specific populations of tumor-initiating cells in medulloblastoma, and that the reduction of SPY1 levels will lead to increased sensitivity in therapy utilizing synthetic CDK inhibitors. To address my hypothesis, I'm manipulating the levels of SPY1 in patient-derived medulloblastoma cells to assess their stem-like properties and changes in response to therapy. For this purpose, I am utilizing an established zebrafish model which constitutes a high-throughput drug screening platform. Manipulated brain cancer cells are injected into the zebrafish yolk sac prior to the establishment of an acquired immune system. Thus, I can observe human tumor formation in the zebrafish and analyze SPY1's role in the process. Thereafter, by applying different therapy compounds, I am able to assess the effectiveness of specific drugs on these tumors in the presence or absence of SPY1 in vivo. Why is my work important? Well, first of all, the results obtained from my study will shed light on the important molecular mechanisms which play a role in the maintenance of the stem-like, aggressive, and resistant properties of medulloblastoma. Secondly, I will try to determine if the targeting of the essential drivers of those mechanisms can potentially lead to improved design of therapy and better clinical outcome in patients suffering from medulloblastoma.